So good morning and welcome to our, this is supposed to be kind of an open mic session to talk about iNaturalist. So possibly a bit of a departure from most of the sessions that you've been to. And thank you for coming. I'm Steve Young. I'm a retired EPA person. And after I retired from EPA, I went through the training and became a master naturalist. And as part of that process, I learned about the iNaturalist app and kind of went crazy. I could claim iNaturalist changed my life. So I'm going to talk about it a little bit, and I'm hoping we can do general sharing, and be very informal and dynamic. I will ask, please go to the notes document, which I hope you can find through Skeds, and sign in. I'd appreciate that to know who we had. And also we have at least one online person. Yes. Welcome to you. Thanks for joining. So let me go ahead and start. I had to do the JBS Haldane quote, so I put that in here, as you can see. Wonderful quote. Yeah. All right. So I thought I would start. I want to set the tone a little bit that this is intended to be, lighthearted, hopefully a good thing for Friday. So I've already had a couple of misadventures at ESIP meetings that kind of track with iNaturalist. So here we go. 2 years ago in Asheville, North Carolina, I was sitting in a session and a gentleman whom I know tapped me on the shoulder and pointed out to me something that was crawling along the carpeted floor. And as I watched, it climbed up onto my shoe and proceeded to start climbing up onto my pants leg. And so I like beetles, so I wasn't bothered at all. I thought it was pretty. At least I thought it was pretty. So, but I did do the Free Willy thing. And now this is, there we go. After taking initial pictures, I did Free Willy and I took it back outside and released it. I took the second picture there. But then I pulled up iNaturalist. I had the pictures on my phone. And I did an iNaturalist observation of the beetle. And I learned, I couldn't have told you what it was, but it was a Virginia metallic tiger beetle, Tetraka virginica. So that was kind of fun. And I told Dave Jones, who had tapped me on the shoulder, I was telling him about it, and I got pushed, by other people in the room. I was trying to be quiet, but I don't have a very quiet voice. So I was embarrassed, but it was fun. So fast forward to this meeting, and I was sitting on my hotel room balcony in the wonderful heat Monday, whatever day that was, Monday. No, it was Tuesday. Anyway, I'm sitting on the balcony relaxing, and I admit I was having a beer, and there was a commotion, and I found a visitor had decided to descend into my glass of beer. So after some commotion, it suddenly was still, and I retrieved it out of the beer and took the second picture here for scale. I believe it was not dead, but it was definitely passed out, unsurprisingly. And so once again, I did an INAT observation, and yeah, it was an American cockroach, which they are native. Some people call them palmetto bugs. Oh, okay. So there. So they are native, I guess, and well, at least they're not German cockroaches. But I was outside on the balcony. So, but anyway, so that's just sort of my lighthearted introduction. That when I get the chance, I try to do INAT observations. So just a few thoughts before we actually move into the open mic segment. I do think INAT is fun. And that's not a bad thing. And it's sort of a gateway, I think, for a lot of people to begin to learn more about biodiversity and maybe even data too, like people who may not be data oriented. So that the learning opportunities that it provides, I've learned a lot more about flies in particular, thanks to iNat. And I now know that flies are more diverse than I realized, and a lot of them are pollinators. Most people don't know that. And there's carnivorous flies. So anyway, to me, the fact that iNat is feeding into a global biodiversity database is like just incredibly important. And I looked, I think the number of observations is over 330 million now in iNaturalist, and over 4 million people have contributed observations. So it's a big deal. But I think interesting for our community, there's tremendous potential for unanticipated uses of the data. And I think we are already seeing people going through the data and deriving other information. More than 6,000 published papers have cited iNat data. That's a lot. And they just put out a little newsletter. It's in the resources about someone who harvested iNat data about bunchberry. It's one of those low-growing dogwoods. And it said, it's common. So you might ask, well, what's the point of putting in observations of this common, organism? But someone began to look at the observations and notice the insect feeding damage on the foliage. And it led to a research project. And they learned that insect herbivory on bunchberry did not appear to affect fruit production. But it was really the iNaturalist data that, sort of enabled the idea for that study and research that they did. A lot of people are doing phenology. They're taking advantage of this rich data set to do phenological work. What are things greening up? When are things flowering? When are they fruiting? So lots of creative uses. And that's one reason why I think, the ESIP community should be interested. And, the possible interaction with a lot of other science data and training models, et cetera, et cetera, et cetera. So those were just a few thoughts. And I did want to mention GBIF, the Global Biodiversity Information Facility, gbif.org, which everyone should know about. But GBIF has billions of data points. It does harvest iNat data that are research grade data. So when you participate with iNat, if your observations reach research grade, then they actually get brought into GBIF eventually. So that's cool. That's the end of the slideshow. I do want this to be dynamic. I should have asked, does everyone feel like you're at least, passingly familiar with iNaturalist? I was kind of guessing that I didn't really need to go, down to the basics, that, you know the basic deal and, about research grade. Hopefully, that's an important thing. But let me just pause for a second. Good question. Obviously, that's been a big question mark in people's minds, is quality of the data. The way that works right now is if anyone else comes in and agrees with your ID, or basically if you have two agreed identifications, it's considered research grade. Is that perfect? No, because some rando person could agree with your observation that's with your ID that's incorrect. But it seems to work pretty well. And because other people are looking at it, there are a lot of specialists who are looking at the data. And so it's imperfect, but they're tending to clean up a lot of it, especially if it is something more unusual. It appears to attract experts who will take a look and weigh in. And there are some people, they don't do observations, but they do identifications. And there's some people who've done, well over 100,000 identifications and brought things up to research grade. So, so there's an AI backend. That's how I didn't explain that, but when you do a photo and submit an observation, you could say, what is this, basically? And the AI backend will suggest what it is. And it is pretty good for a lot of organisms, but, clearly spiders as an example, lots of insects. To get down to the species level, often you may need to literally dissect the organism, So, but you may be able to at least idea that, genus or family level, which is not without value. So I've heard them advocate, try to take it as far down the taxonomic tree as you're comfortable with. And if you're not comfortable beyond it's a monocot or a dicot, that's not the end of the world, because someone else might look at it and bring it closer down to species level. And I should have said, I would call myself an enthusiast. I'm not an expert, and I'm not really properly trained. I've worked on bioinformatics stuff for a long time, but I lack formal training. So feel free to call me out if I say something that doesn't sound right. But any other questions? Yeah. Oh, yeah. Thanks, Ben.
I was going rogue. I would love to use any and all GBIF data that are available, including iNaturalist data. Something that I'm curious about, and Steve, you may not necessarily have the answer, but maybe someone in this room has the answer, is how certain we can be about a location of an observation when someone's making an observation from like a cell phone. I don't know, can we assume that observation is within like a 5-meter radius, 10-meter radius, 20-meter radius?
Yeah.
What is sort of like the uncertainty around that observation point?
Right. That's a great question. And answer is complicated. If you have location turned on, if you're using your cell phone and you have location turned on, it's probably pretty good on average. But depending on terrain, possibly tree canopy, how well it's picking up GPS. If you can see, you should be able to see the photo metadata. So if you really want to know, you may be able to look at the metadata and get a sense of, okay, it was capturing, location, and it's showing in the actual metadata for the photo. But there is a lot of concern about, for rarer organisms, concern about, you don't really want to publish the exact ID, especially if it's threatened and endangered. My understanding is that they are now obscuring the location if it's a listed threatened and endangered species. So, okay. Yeah, please.
I'm going to confirm that what Steve said. We'll verify. Yes, and it's not just for endangered. I think it's threatened or even rare species. Can I share a little anecdote? Is that appropriate? Okay. I like to hike around, and I'm often in the back of the hiking group because I'm squatting down to look at a little thing or a flower and putting it on iNaturalist. And one of those things that I had stopped to photograph on the side of the road ended up being a stream orchid. I had no idea what they were. I put it on iNaturalist, and then I was getting frustrated because I couldn't see the location of it. And then someone reached out to me, and it ended up being the director of the local Naturalist Institute. And they were extremely interested in where this observation was, because it hadn't been seen in the Lake Tahoe Basin since the 40s, 30s or 40s. And so we ended up working together and organized the community walk to go look at this stream orchid on the side of the road. And it was pretty fun. But yeah, to the point, it's the location was obscured. And I actually got some other requests from orchid enthusiasts to know where the location was, which was kind of an interest. I didn't really anticipate that or know how to deal with that, but yeah, there are some protections in place.
Yeah, this is exactly what I was hoping for, that we just had some interactive. So let's keep going.
So there are ways to there are ways where it will show up as an obscured location, but the actual location data is in the iNat database, and you can find it, and you can share that with a particular project or group. So if there's the Orchid Research, legit Orchid Research International Association, if you subscribe to that group can be allowed to see an obscured location so that you can get that information. Also, when it comes to locations, you can put uncertainty in there as well. But some of that is probably more on the user, not necessarily what everyone's going to do. So somebody who's really invested in how their observations may do that. There are other problems in my disclaimer. In my non-professional activities, I work with the North American Mycological Association, and we put on a bunch of forays where people bring in mushrooms from a bunch of field sites and they get identified. A lot of times they get photographed and documented at the place that they were brought in, because that's where the photo metadata is from. So sometimes you will see a giant pile of observations on top of each other that could be from the surrounding 50 miles, So yes, there is absolutely a lot of location uncertainty that you have to be comfortable with. Now, is it going to be 2 states away? No. But it's not necessarily going to be 20 meters, but it could.
Yeah. And I think that's a good point, that as a user, you can make your own call. And so you can choose to obscure the location or make it private. You can do that as a user or as an observer. Sorry, I should say. But if you're a data user, it can be complicated for sure. So let's more. Let's.
I guess it's in the same vein. So my other question is related to these responses. When those data go onto GBIF, they have data flags like fuzzy taxon rank and fuzzy location. What does that mean to you? Great.
I guess that sort of a county level location is not necessarily any more accurate than a historical herbarium specimen, because you've got a description typically from before a location description from before the modern era, before GPS. I mean, modern herbarium specimens may have a precise GPS location, but most of your historical records have a near Austin, or like north on Highway 2. So it's sort of
exactly.
I don't think any of our other historical biodiversity data is necessarily any more precise than maybe the modern iNat is. And that's the sort of level of fuzz we need to be comfortable with.
Does fuzz mean like so I'm wondering if fuzzy means like we're not really sure, it's kind of fuzzy, or like we fuzzed this for the protection of the orchid, for example. Is it both? Maybe this is a question for GBIF folks.
Yeah, I don't know the answer to that. Yeah.
Okay.
I feel like in many cases, of course, it depends on what you're trying to do, but often it's going to be a point of departure. Like it might lead you to realize, I want to go to this general area, or I want to dig into other data sources and see if I can find some, maybe old papers or whatever that might help me pin it down more. I had a big argument with a friend of mine who's kind of a spider enthusiast, and he and his I'll call her mentor, were very dismissive of iNaturalist circa 5 years ago or so, and they were basically saying its IDs for spiders are garbage, and so in effect they were saying so it's really useless. And I counter argued, well, what if you've got all these citizen scientists out there roaming around, and what if they submit a bunch of observations that are geo-referenced, and you see from the photos, well, they're finding some interesting spiders, and this is a location that we didn't know. So we're going to go out to that location and do professional, collecting and figure out what these spiders are, and actually, collect specimens and all that good stuff. So I think, and they've come around now. They are no longer dismissive of iNaturalist. And I think they're seeing, well, we're introducing a new audience to, biodiversity, including spiders. And that's kind of a net positive,
Yeah.
I should be asking people to do like a quick intro.
Oh, yeah.
Please. I should have said that.
You want to?
Oh, sure. I'm Kelsey Gilsman. I'm a community fellow and also a postdoctoral researcher at NASA. And I have been wanting to use GBIF data on this, pairing GBIF observations with satellite data and, maybe for ground truthing, but I want those observations to be on point, literally.
Mm-hmm.
Joshua Berkovac, Oak Ridge National Lab. A lot of my iNaturalist stuff I'm talking about today is not professional and does not represent Oak Ridge or the DOE or blah, 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 blah. Though we do use it sometimes in limited senses in my professional career. So if that comes up, we'll talk about that too.
Cool.
I'm Don Haas from the Paleontological Research Institution and its Museum of the Earth and Cayuga Nature Center and Center for Climate Change Education, all part of our organization. And couple of thoughts. One, my colleagues and I have done a lot of work on virtual field work with mostly K-12, but K-16 really. And one of the things about things like iNaturalist is it flips the problem on its head of technology keeping people inside and uses technology to draw people outside. And I really like that. And I added to the notes document a link to our BioBlitz page. We do quarterly BioBlitzes using iNaturalist for the Cayuga Lake watershed region. And just last weekend was the summer one when we run them from Friday through a Monday, so that the general public and kids in schools can participate. And I think we've been doing them quarterly for maybe 5 years, maybe a little longer than that. And we started doing them writ large with a big BioBlitz maybe 10 years ago at our Nature Center in collaboration with a whole bunch of scientists from local colleges, particularly Cornell. I think there were over 100 scientists involved in a 24-hour BioBlitz, and the quarterly BioBlitzes are nothing like that scale. And anyone who who uses either iNaturalist or Seek, which is also from iNaturalist and sort of the more kid-friendly version of iNaturalist, those observations feed into the BioBlitz if they record the observation anywhere within the watershed. And did I say I added the link to that to the resources page?
And that's a good chance to remind people, if you didn't sign in on the document for the session, please do sign in. And speaking of BioBlitzes, let me just do a quick commercial. There is something called City Nature Challenge. And who knows about City Nature Challenge? Not everyone. So it's a really fun global event now. held every April, late April, if I recall correctly. So kind of not far off from Earth Day. And I think it was over 600. It's not necessarily literally cities, but, kind of geographic areas, metros, whatever. And it's like a friendly competition for which area can collect the most observations, the most observers, the most identifications, the most species. And it's really quite a lot of fun. And a lot of people go out it's a 4-day period for collecting observations. So a lot of people, it extends across a weekend, a Friday through a Monday. So it mobilizes a lot of people to get out there, and great learning, educational thing. The friendly competition aspect is kind of fun. So that's something to be aware of. You can search City Nature Challenge and track that going forward. And the whole BioBlitz thing is but they're closely related to each other. Yeah. Oh, okay. Yeah. Please do make sure. Yeah.
So hi, Ben Galuski from the National Center for Supercomputing Applications at University of Illinois. I now feel self-conscious about this question when you said Seek is more kid-friendly. My question was about the taxonomy of the iNaturalist app space. I first started using it actually hiking around in the woods behind the Burlington, Vermont ESIP meeting.
Oh, cool.
And it seemed like iNaturalist is a different app than Seek. I settled on Seek for use outside.
Closely related, and I believe they're using the same backend that does the pattern recognition, the AI stuff. But Seek is interactive in a way that baseline iNat is not.
Okay.
Also for kids, Althea mentioned there's that sort of social media aspect to iNat where you can hear from people commenting on an observation. I learned that the schools are very concerned about that the social media connection could lead to abuse. So a lot of the schools will use Seek, but not actual iNaturalist out of privacy concerns.
No, it's like Seek and Find. And
yeah.
And so one of the things, I don't know whether iNaturalist does this because I've only been using Seek now, is that it runs. Okay, because it mostly, okay. Not just for kids, but one of the things I find so impressive about it is that the model has actually been downloaded to your phone. And so it'll recognize plants with no internet connection.
Oh, I didn't know that. Interesting. Also, if you're using Seek, you have the option of telling it to turn it into an iNat observation.
I think so. And I
have not personally done that, but I think so. Yeah.
Okay. I need
to. It's totally cool to use Seek, but remember, it is nice to build the database. So at your discretion.
And I'm usually using Seek when I don't have internet access, and so all those buttons seem meaningless. One of my favorite things, because Seek, you can sit there and keep moving your phone around until it recognizes what it's looking at. And one of my favorite experiences was doing that with my poodle, and it settled on carnivore.
By the way, iNaturalist does discourage people. That they basically say, please, no humans, no dogs, no cats. But they're trying to keep people from submitting garbage. Understandably, why don't we? What? And then let's do you, Carla, and then I want to give Althea a chance to introduce herself and probably talk more about what you're doing in your interests. But
I just wanted to make a quick comment, probably that you might think about with when it comes to the geolocation data quality and research grade, is that anything that's marked cultivated or garden or non-wild is excluded from research grade, because that's obviously a big problem when it comes to look at this ginkgo tree, in New York City. So anything cultivated or non-wild does not make it onto GBIF.
So I don't mind displaying my ignorance. I've used Globe Observe. Oh, I'm Carla McAuliffe. I am organizationless right now, it's just me. But I'm a former K-12 teacher now, working with teachers and conduct educational research. Anyway, I've used Globe Observer a lot and use that with kids, understand its interface. Never used iNaturalist or Seek. So but I'm going to now. When you use those, do you get that map interface also? Do you see your observation on a map? You do. And then what you were saying, creating a database that's like your own of all the observations you've made. Do you have a way to?
You can. So they actually have some pretty powerful filtering capabilities. So by applying filters, you can filter to only see your own observations. Okay. And then you can play with, well, I'm only interested in my plant observations or whatever. So the filtering actually gives you some pretty powerful. It's you have to learn it a little bit. I'm like a total novice at taking advantage of the filters, but they also allow you, they have some really good webinars that I put a link in on the document to their tutorial videos. But you can, if you're interested in a particular taxonomic group, you can filter to only look at observations for that group in a particular location. Mm-hmm. And you can look for observations that are not research grade. And if you like, you're not really out there roaming around, but you do have some expertise, you can find observations that need identifications and go, okay, well, I know what that is. And so you can help build up the quality of the database by providing IDs for organisms.
The other thing I was going to mention is that from what Kelsey said, I have an opposite interest in the fuzzy data. I'm putting together a proposal of teaching about uncertainty, recognizing uncertainty in science for the K-12 community. So this is opening up a window because we have now one line left of space in the proposal. So if I add something, I'm going to have to take something out. But what you were saying and just some of it might be really worthwhile to go into, what was it, GBIF? And, have those kinds of conversations about where the data came from that we don't know. It's fuzzy. So anyway, thank you for that.
I have to disclose, I'm very biased about GBIF because I helped in the creation of GBIF back in the day. And so, I feel like kind of a godparent or whatever to GBIF. And, but yeah, I mean, I think GBIF is amazing. And so it's a huge, huge biodiversity resource. A lot of it is museum data. So where you're actually tracking two type specimens sitting in museums, herbaria. So it combines field observations. It also harvests from eBird. For those of you who know eBird, GBIF does. So you're bringing together observational data and actual specimen data, for biodiversity specimens. So it's amazing, I think. Okay. Althea, you want to intro, and if you want to chime in based on what you've been hearing so far, talk a little bit about what you're doing, what your interests are since we chatted.
My name's Althea Marks. I am a data scientist who works with seafood trade data at the University of Washington. My background's in marine ecology, environmental science. So looking at and documenting plants and animals is still in my personal life, like I mentioned, but mostly just been for identifying flowers and insects in a personal capacity. But recently, I think this is probably what you're alluding to, the purple loosestrife. Yeah. So I live in South Lake Tahoe, California, and that is on Lake Tahoe, which is an incredible place, and there's a lot of environmental organizations there focused on preserving the watershed for the lake. It's a huge economic force in California and regionally. And so there's a lot of focus on invasions of Eurasian milfoil, curly leaf pondweed. California is all up in arms about possible golden mussel invasions, etc. So it's really a leader in as far as aquatic invasive species. However, just going around on the trails in my neighborhood, I live next to and on top of a dredged marsh, which is absolutely terrible. The worst thing that probably ever happened to Lake Tahoe. Anyway, it's disturbed. And there's a lot of non-native species in there. But then I noticed this really beautiful purple flower. And of course, I pulled out iNaturalist. I was like, what is this thing? And I was like, oh no, you've got to be kidding me. It's purple loosestrife, which my first job as an undergraduate was working with aquatic invasive species in the Adirondacks and in upstate New York. And one of the species that I learned about was purple loosestrife. And if anyone, I'll just give a quick little purple loosestrife introduction. It's a wetland invader. It's highly documented. It's native to Europe and Eurasia, I think. It's extremely effective. It'll form dense stands and exclude native species, impacting wildlife habitat. And I don't actually know the evidence for water quality, but anyway, highly prolific seed production, classic noxious weed kind of scenario. Turns out no one's paying attention to it. And I've been sending countless unanswered emails. I've been showing up to board meetings. And everyone's like, oh, did you go to the league? Did you go to the RCD? They're handling it. They're doing it. I'm like, no, they're not. Actually, it's not on their weed list. Like I went to their weed polls for the meadow wetland meadow restoration, and it's literally not on the pamphlet they're giving out. So I don't think anyone's paying attention to it. And I was like, how do I document this? How do I share this? I can be a crazy lady around town about this purple flower. But so I decided to be a crazy lady tromping around in the wetlands and taking lots of photos and posting them on iNaturalist. I was like, there's got to be a way to consolidate these. So there's an ability to make a project, which is, I think, a lot of the...
Oh, yeah. We didn't talk about projects.
Yeah. And there's a couple different levels and they behave a bit differently, but I made a classic project. Steve, is it, could I just put it on the screen? Okay. To Carla's question about the filtering, this is the landing page for the explore function. So you can filter down through here and just like, zoom into whatever observation. And there it is. But anyway, this is my page. Oh, I'll stumble all over the place happily. Oh no. So it was a very easy little template, and I've got this basic little web page that I love. I got to pick the color to match the purple loosestrife. And I went crazy. So I've got 314 observations in here. And there's a handful. So basically there's a few constraints that I was able to do. And I was interested at limiting purple loosestrife observations globally down to my city. But what works most is I think it's in the watershed. So you can constrain it in a couple different ways. And I haven't gone in depth with that. The watershed was fine. And then I got to choose the map representation. And I think, let's just refresh. Okay. So yeah, I think, I'm not sure how it chooses the default, but you can change the different map overlays. I actually found that the points lined up better with the satellite. But anyway, let me zoom out so you guys can see. There's Lake Tahoe. It sits right in the bend of California and Nevada. And there's the bend right in the middle of the lake. And down here, this used to be one continuous wetland, which was the filter for the main tributary or input. I'm losing all my words. Water input into the lake. And then, in the 50s and 60s, some developer had a fantastic idea of making this like Oh, it's terrible. It's really, don't get me going. Anyway, I tried to do my little part by catching bullfrogs. And anyway, here's this back water. It's called Talac Lake. Anyway, here it is. And this is super disturbed. Like there used to be like this water facility here for the HOA. And you can this was actually just fill in here. There's beavers. It's, you know, it's got potential, but I've been going through and documenting each one of these, and you can see, yeah, the position is off a bit. Okay, I will. Let's just click on this one. Yes. Oh, I didn't, you know, I usually take higher quality and multiple for my observations, but because I was documenting so many, I just went for quantity. Yeah. Oh, yeah, this is a terrible one. But often mine have my hands in there too for scale and to help the camera focus. Yeah, okay, so this is a decent one. Okay. Yes, but I solicited their input, which is a bit unusual. Like I have observations that have been up here for years, and I get a notification in that like somebody has added to it. So like I'm continuing like old observations, someone's reaching out to it. Anyway, yeah, people can find it if you're. Well, this one in my case, I'll show you. I went to Purple Loosestrife, which each taxa has its own landing page. Here it is, and you can see top observer. So this is the observer is the person who's posted the most observations. Top identifier, and so this person has 9,555. They've contributed by confirming or denying identifications. They're just looking at the photo. So I actually went to, where did I find them? I actually can't remember how I did it. I thought it was on this page, maybe not US.
There's so much data, it can be a little bit overwhelming at times, you know, but that's a good thing. It's a rich resource.
Yeah, it's fairly easy to navigate if you spend a little time with it. Anyway, there was a list. Somehow I got to a list of top identifiers for Purple Loosestrife.
Oh, yeah.
And I was like, well, I'm the crazy lady in town taking pictures of the Purple Loosestrife, and I needed some support that this was in fact the species that I was concerned about. And so I went to this list of top identifiers and I messaged them directly. Like, here they are. Like, yeah, this, you know, I said, hey, my name's Althea Marks. I'm a community member here. Like, please, I'm concerned. Okay, this is probably a really terrible, I spent like 3 minutes writing this out, but I was like, whatever, I'm just gonna send it. Like, please come help me confirm or reject. I'm pretty sure this is Purple Loosestrife, but I see you've done a lot of identification. I would really like your help. Here's my project page. Oh, this person couldn't do it because they were very busy with another taxa identification group, which I just loved. Like, you're excused. Yeah. But I messaged the top 25 people, you know, and some people were like, absolutely. And those are the ones that helped bump my 300 observations up to research grade in a week, when it would have probably taken years otherwise. So it is a community.
There's also a phenomenon. I do a lot of killing invasive plants. I'm fairly good at it. And we've discovered over the years that there's a lot of crazy people out there kind of doing Lone Ranger work on invasive plants. And the people are finding each other now. And so iNat is one of the ways that, so there's a concept called early detection and rapid response, where for a new outbreak of an invasive, to try to like catch it early, because in many cases, it's usually plants, but in many cases, if you catch an outbreak early, you can actually eradicate a localized outbreak. And so I'm seeing people taking advantage of iNat to find the early detection of an invasive of concern and then mobilize. John, we'll get back to you. Yeah, let's keep passing the mic because we have a few people that haven't introed yet. So
hello.
Yeah.
Hi, everyone. Yeah, I'm Sean Carter. I guess I'm at Leidos. Again, this is not my professional capacity, but at one point I was a bird biologist and I did a lot of distance sampling. And then I actually wrote my dissertation on EDRR, early detection and rapid response. And my question kind of really, and now I'm just a corporate slave, but my question, it sort of relates to this whole conversation where iNaturalist, eBird, etc., these citizen science platforms are what we know in the species distribution modeling community as presence only modeling. We don't put zeros in our observations. And I'm curious, like, what's the link? Because I, as someone who did put a lot of zeros in a lot of databases professionally, like, and I was paid to do that. What's the link there? Like, how are these platforms actually integrating with species distribution modelings, maybe invasive trajectory modelings, and
so I'm going to make a crazy response to that. Based on what we're seeing, especially with higher resolution remote sensing, at least in the case of plants, I think we're getting closer to where if you now know that you have presence in an area, you can hypothetically, and believe me, I'm sort of talking beyond my knowledge base, but what's the spectral signature for that plant? And then, okay, now we know we want to look by area of interest, take advantage of remote sensing to look for the signature and be able to more exactly map distribution. I think there's just a lot of ways to interface with other work that's like what I call mainstream ESIP type stuff, and definitely modeling distribution, where the iNat data are helping you train your model. That's still not directly addressing abundance, but at least like refining the distribution aspects of the
Can you facilitate those relationships with scientists who know those methods, such as spectral and mixing, et cetera? Maybe you can. Yeah, here you are.
I also have thoughts on that, like presence only situation that I kind of forgot about when I came in with my fuzzy question. But so actually, I would say eBird has one of the best approaches to dealing with the presence only data. The way that I have navigated the data that are from iNaturalist on GBIF or any other data on GBIF is actually, so with plants, using something like the National Ecological Observatory Network that does do these exhaustive plant surveys, and using that as like a standard for comparison where it's available. That's one approach, is to sort of like verify and add. But another way that I've seen a lot of people navigate it, and the way that I would navigate it, is to consider the completeness. So like you have these exhaustive surveys of vegetation from NEON. I would call that like it's complete. They recorded all the plants that were present in this plot. Whereas an iNaturalist observation is like, I saw this plant here, and that's a fact, but they don't record any other observations. That doesn't mean there's nothing else there. Yeah. And so, right, yeah, it tends to be close to roads, charismatic, they're biased. So there's a lot of like ways to correct the biased data, or, biased data. that others have done. But the way that I think about it is like completeness, and the completeness can be in terms of, how many observations are in this space compared to how many observations should be in this space. Or how many taxa did they see compared to how many taxa did NEON see? But also it's possible because NEON is doing these exhaustive plot level surveys, but the plots are not covering every square foot of their land. There might be observations in GBIF that are providing something novel that's not in the NEON data that really is there.
Yeah.
Right. But the way that
collections where the specimens were collected, 50 years ago and housed somewhere, and GBIF has access to that.
Yeah. But yeah, completeness is sort of my approach to sort of correct for the lack of observations. And so we can say, yeah, this is here, but take with a grain of salt what's not listed, because it's like 99% incomplete.
Yeah. So. This also brings up, there's some fun things happening with audio monitoring. So like a lot of you probably know about Merlin, which is also so much fun. And it's another great gateway for people to get into birding. I'm seeing that a lot. It's really fun to watch. But audio monitoring in general, a friend of mine has monitors deployed near Denver, in a natural area. So they are recording everything. And there's software now that it's kind of like Merlin, but it can cover, if there's an audio signal from the organism, you can tease out, lots of species, calling amphibians, insects, birds, bats. And so that starts to give you some kind of rich data potentially when you're starting to bring in the audio side too. So that's just an interesting developing area, I would say. I want to hit a couple, or not hit, touch a couple other people who haven't introduced themselves. Yeah. So, Don, did you want to say something?
Yeah. So sure. Just one thing two things on purple loosestrife. I live in upstate New York and I've lived in Michigan. And the populations are way down, and I think that has to do with species-specific beetle that was introduced for control. Yeah, but that is good news. It was very pretty when they were around all over the place in the 90s, but it was also pretty invasive. Thinking about some of the advantages of iNaturalist, I have an undergraduate friend from upstate New York who now has a place on Lake Tahoe. He lives in California on the coast most of the time, but has a place in Tahoe. I don't know if it's South Tahoe, but I'm pretty sure he does not vote in a way that supports us. But I think if he loves Tahoe, and I assume that he does not want to see it overrun with an invasive species. So I'm going to talk to you about inviting him to your project so that he's getting out there and collecting data and maybe thinking about, maybe not letting it run wild. And I think that's a nice connection for citizen science stuff.
Yeah, that it's cool the way that iNat is facilitating these connections, and so unexpected, the serendipitous, people finding each other and realizing they have shared interests in, organisms, whatever. Yeah. I'm going to let you two introduce yourselves, please. And also any questions, comments?
Leah Coloswano. I work with the USGS, and I'm just here because I'm interested in iNaturalist and have been dabbling. So it's fun to hear you guys talk about it. Thanks.
Okay.
Yeah, Sean Thibodeau, also USGS. Sorry, I had to run out for a second. So I missed the last 10 minutes of this, but full disclosure, I was super into these tools a couple years ago, kind of fell off, but getting into some more like data visualization stuff with my work. So, really dense networks that we're providing dashboard tools to our cooperators. So kind of trying to get back into like the citizen education side of the data we're collecting.
Cool. And
hi, I'm Lalita Kempka. I teach at middle school, junior high school students. And we are going to be working on, hopefully, if I get accepted to the program by NASA, Growing Beyond Earth. It's a project for junior high students, and I'm learning about iNaturalist and trying to teach this to my students.
Cool. I hope you're finding this informative. Nice. And our fellows, we don't want to leave you out.
Thank you. Hi, everybody. I'm Gabby Brown. I'm a first-year PhD student at the University of Oklahoma. I don't have background in any of this. My background's in atmospheric sciences and meteorology, but I do a lot of taking pictures of flowers and vegetation and stuff. I have a whole photo album on my phone, and I love birding and watching. I have a bird feeder at home that takes pictures of the birds. And so this is completely a hobby, and I had no idea that iNaturalist existed. So it's so cool to kind of be able to, moving forward, contribute to science.
Well, that brings up a good point. If you've got a photo roll sitting around, it doesn't have to be prospective. You can retrospectively go back to your photo roll and upload and create iNaturalist observations. So there's so many things we could have talked about, but Althea is bringing it. So it will ask you for a location if locational metadata isn't already attached to a photo. But if you it's not too difficult, and you could, this gets back to the earlier question, you can at least give it an approximate location. You're basically drawing a circle and get, ballpark as close as you feel comfortable. So, I know someone, a professional naturalist, who had thousands of, a lot of them were film photos that, he digitized them. And so he's taken thousands of old observations and turned them into iNat observations. Now, it is important you want date and time. So knowing him, he probably had logged the dates of the photos. The time is less important, but that was a huge amount of work. I can't believe he did it, because he's got like 40,000 observations in iNaturalist. But I think he's into it, right? Because, he's proud of what he's done. And it's a lot of cool pictures, cool organisms, but now it's in the database. Bobby.
Yeah, I'm Bobby Saba. I'm a community fellow, also a PhD student at OU. Also a meteorologist, so not really my work per se, but I would say that Gabby takes pictures of plants and all that kind of stuff for the two of us. So plenty of photos lying around. But this is super cool. This is really neat. I think, to be honest with you, I'd never heard of iNaturalist until about an hour and 3 minutes ago. But yeah, super excited. This has been really cool, and hearing about all your passion work. I don't know if we want to bring it over to Zoom.
Well, and you all are reminding me. So I am a weather geek, and okay, you've got weather radar, right? And so weather radar is picking up some interesting biological data, as I know you all are going to be aware. Like, it's clutter or noise to meteorologists typically, but you can see on weather radar the dispersal, for example, of birds and bats often is visible on weather radar. And there's this amazing work going on now to track bird migration. I think it's called BirdNet. Taking advantage of weather radar and also modeling, you can forecast big migratory movements of birds. And then you can anticipate most of the birds migrate at night, and they call it fall out. They fall out and roost, they rest during the day. So birders want where are all the warblers going to fall out? When is there going to be a big migration? Where are they likely to fall out? Because that's where I want to go, because I want to see a whole bunch of warblers during migration. So that to me, that interaction with radar data is just okay. Okay, yeah, thanks, Carla. I had a feeling. So what Birdcast does is fascinating. And to me, I love that kind of interconnection between, like who would have thought, and even insects, weather radar insects too. So sometimes you can see, I've also learned, and we're learning, forgive the babbling, but we're learning more about insect migration. Like everyone knows that monarchs migrate, right? Although I don't think the average person doesn't realize it's not the same monarch going all the way, from the summertime down to the wintertime. It's multiple generations, but it's still a kind of migration. Well, a lot of dragonflies migrate. I only recently learned that. So you also have some dragonfly migrations and other insects too. So, so many cool things going on. Let me get Joe and then we'll circle back. Oh, good reminder. Okay, Joe, just to introduce yourself, please.
Hey, I'm Joe. I'm one of the community fellows. I've also used iNaturalist a bit in the past. I'm a geologist, not a biologist, so all the stuff on the surface here, it's just kind of like pond scum to me. But it's still nice to look at. But I've used iNaturalist, especially in helping with some. I do look at birds every once in a while, and it's helped with like distinguishing. I'm terrible with shorebirds, so like your sandpipers, and like ended up finding that I wasn't looking at what I thought was a sandpiper, but like a little ruddy turnstone, which made sense because it was walking around turning over stones, and I was like, that's interesting. That's where the name comes from. So, yeah, I've appreciated it.
Well, you see a lot of lichens growing on rocks too. Okay, maybe our online person, are you unmuted? Can you introduce yourself, please? Elizabeth? You're showing as muted right now. Elizabeth. Yeah, go up.
Try again. Can you hear me now?
Yes.
Oh, sorry.
Yes.
I'm so excited to find my people. I'm the weird, crazy person here in my area as well. So a lot of people don't even want me to teach their kids because they're afraid that I will turn them into tree huggers. And some of them become tree huggers anyway because they sneak down to see me. So I live in a real rural area of Georgia. And yes, I've already known about iNaturalist, but I was really excited to be able to show her page and show that to my students. So anyway, I teach 6th grade earth science, and I still try to branch out, and I'm learning about new careers just listening to this and being part of this week so that I can introduce all that to my students. So that was my interest in this.
That's great to hear, Elizabeth, and welcome. We're glad you could join us.
Thank you.
Okay. How about we go?
Is this still on? Yeah. Okay. So I just want to be optimistic here. And I'm actually very excited about this because I invest a lot of time and energy in the Globe app, and it looks like that's going to be sunsetted. Thank you, NASA education funding. Not, but the citizen science, I've never been afraid to teach kids actual scientific tools or tools that, like in 2006, I taught kids and teachers how to use GIS when it was very clunky. There's no reason to be afraid of something that a scientist would use. Now, I know this comes out of the citizen science world, but it makes science very accessible. I still want I'm caught between, I want to teach kids about uncertainty, but the data that I want them to collect, I want it to have the actual latitude, longitude, because I'm back to being a GIS person. And here it is. Here's as many attributes as you can get about this thing, so you can map it in different ways. But the hope for me is this is really great. Nobody can shut this down. They can't say you cannot use iNaturalist anymore or Seek. So this is, I think, hope for the future in terms of continuing to get data with understanding the caveats about the data into the hands of teachers and kids and have them collect data. So that's what I wanted to say.
Cool. And let me interject for just a second. iNaturalist is now a standalone non-governmental organization, NGO, iNaturalist.org. And they do rely on donation support, to operate. So donations are encouraged. I have no, I am a donor. So, but that's just something to be aware of. But it was originally developed largely out of California Academy of Sciences, but they did turn into a freestanding NGO within the last, I think, 5 years or so. So they're now reliant on public support.
And so I am wondering if you can look at iNaturalist and change over time. So, watch the purple loosestrife move in and my mind is racing thinking about putting things together. So yesterday outside of Albany, they got almost 12 inches of rain in 24 hours, wow, which is unheard of. And I think and I learned yesterday that USGS has stream cameras where you can go and look at, and you can look at any time period that the camera has been there and do a time lapse of it. And so, Carla and I have that link if you want to check that out. But go to what's the name of it? Do you remember?
USGS. Yeah. Sean would know.
I'll pass it to you in just a sec. So looking at that and looking at where the Mohawk River overflowed its banks and tributaries to the Hudson around there, and seeing what was there the day before yesterday, and watching it what comes back, and doing the same sort of thing with fires and looking at fire maps over time, and using the time-lapse feature in Google Earth for construction sites. And, when you put in a solar farm, what are you taking out? What comes back? And thinking about all of those possible connections just makes me feel warm and fuzzy and excited about things. So.
Yeah, that real-time camera service for USGS, HIVIS, HIVIS, developed by Keegan Johnson and a couple other folks. So it's Water Mission Area, pretty valuable new service in the last couple of years.
Yeah, so go there, look at Albany, and go just a few miles south, and do a time lapse over the last few days. Really interesting visuals.
Speaking of time, one of the when you look at the species information in iNat, they have a little phenological chart for the species that actually shows you the distribution time of year of observations. So that could be a little bit interesting to, just to see when, the most observations have been made for an organism, what time of year. So you'll see that curve, and occasionally you'll realize it's kind of unusual to see this thing, at this time of year based on what the observation data are showing. A smart data scientist could undoubtedly harvest iNat data and generate like visualizations, about at least the observational pattern over time, put it on a map and show for a particular species of interest. The data are there. You'd have to have some smarts that I don't have to generate that kind of a visualization, but the data are definitely there.
Yeah, I was just going to say it's a little clunky but it's not very hard to get CSV exports filtered however you want, by what species, what region, county, time frame.
Yeah.
It's very easy to get that out. You'd filter to only research grade, filter to only things with pictures, filter it. And then you can dump it anywhere you want, whether you want to spatially map it, look at it over time, look at only a cool thing with a lot of plant observations, depending on whether people use them or not, are observation fields that not only just say it is here now, but you can check whether it is flowering or whether it is senescent, or, so there's life cycle additional observation fields that are used as well.
It's from iNaturalist. You don't have to go to GBIF or whatever you're saying. Just in iNaturalist, you can get a CSV. Okay.
Another capability it has, like we briefly talked about projects, but anyone can define projects in iNat, for your own personal purposes, or, for a group or whatever, and you can define the parameters of the project. So some projects are set to automatically harvest observational data, so it's seamless for the observers. Other ones, the observer has to join the project, so make a conscious decision to join a project. But it also, it's very flexible about creating additional attribute fields. So people research, this is usually actual legit researchers, will create additional attributes to track an observation. So, for example, people will look, the observations are of insects, but what's the host plant for the insect, for example? So you could basically create an attribute host plant, and track that as additional data so you could actually take an observation and continue to add more value by, again, if you're a researcher in particular, you're the likeliest to do it, creating attribute fields that support the research that you're trying to do. And I think that's what they did for that bunchberry project that I mentioned, was that they were characterizing not just that there was feeding damage on the bunchberry in that observation, but how extensive was the feeding damage. And then they were able to correlate that with fruiting production. They did some actual experimentation, though. They used a hole punch to simulate insect feeding damage and then track the productivity of the individual plant later. What are we bringing? And this one, it's also showing, I see, it looks like, there. Oh, right. You can see suggested IDs that were withdrawn. So that, there was a lot of activity on that one. And there are disputes, like you would expect. People are usually civil, but unsurprisingly, people don't always agree about what something was. And of course, often the photos, you are not going to be able to confidently say what species it is, but maybe you could at least be confident, well, we're pretty sure it is genus whatever, even though we can't agree on what species, Yeah. So it's the potential is just vast, and I keep repeating myself, but it's so cool that it has all of these educational values, but also very serious science values, for researchers. So it's a cool thing. Well, we have a few more minutes, so I do before we wrap. I also, if anyone has any recommendations that we might want to try to feed to iNaturalist, I do kind of know one of their main people, and so if there's anything that we've talked about where you think, well, it would be really nice if they supported blah blah capability, I'd love to capture thoughts like that. So, go ahead.
Well, this isn't a recommendation for iNaturalist, but I was going to ask if you're going to lead an ESIP BioBlitz at the next meeting. It'll be a fun session.
That's a fun idea. I will take that under advisement. Someone said someone said we're in Buffalo, and maybe if we're able to tap into some local knowledge.
Tiff Nature Center is probably 2 or 3 miles from the Buffalo Convention Center. And I know folks there, and I'm sure they would be very happy to host whoever wants to come. And it's essentially on the waterfront. It's actually reclaimed brownfield. Part of it's over old industrial stuff. Part of it's over an old landfill. And it is, across the street from Lake Erie. And if you haven't been to the Great Lakes region, they're great. You wouldn't know from standing on the shore for much of Lake Erie that you're not on the ocean. So that's one thought. In terms of what would be really cool to see from iNaturalist would be a time lapse feature where you could highlight an area and look at observations over time. I think that'd be super cool.
I'm curious if anyone in here this was my first exposure many, many years ago, seen AntWeb. Antweb.org. It's the world's largest online database of images, specimen records, and natural history information on ants. And it was started years ago, like many of these, mostly by scientists. And then, but they were very strict about it. It had to be verified by scientists. And so this is a really cool, but I don't know if anybody in iNaturalist is looking at ants, but they might want to go over here because there's a ton.
You name it, and people are looking at it.
Yeah. So those people might benefit from knowing about antweb.org.
There's also Bug Guide out there. I think it's bugguide.net. Or anyway, Bug Guide, and it's pretty strong on insects. So often people, when iNat is inconclusive about the ID of an insect, you could go to Bug Guide and put it out on Bug Guide, and they have a lot of serious entomologists who will weigh in, and often they'll ID the insect with a pretty high degree of confidence for you, and then you can reflect that in your iNat observation. So Bug Guide is a cool resource. Are you pulling it up, Carla? I think it's .net. Okay. So that's worth taking a peek at too. How about any other thoughts? We've got like our final 7 minutes. Of course, Well, it looks like we've exhausted our. I want to thank all of you for coming and participating, and thank you to our fellows for support as well. And I was hoping that this was going to be a fun session, and my hopes were achieved, I think. And the sort of cross fertilization, I think, is super fun too. Like I love being able to bring in, well, there is a connection to meteorology kind of and stuff like that. So it's been fun. I forgot to put my email. Well, my email is pretty easy, frasbo at Gmail. Frasbo, don't ask, it was sort of a made-up word, FRAZMO. It's easy to be. It was available when I set up my Gmail, and I liked it to have a short. But it's been pointed out to me it's not very professional. But I mean, I don't have to worry about that too much anymore. So, all right. So with that, I guess we are concluded. Thanks.